electron donors and acceptors from inorganic salts. And due to this, at the end of the growth or at the end of the metabolic activity, amount of the energy generated is very low as compared to heterotrophic organisms which are utilizing organic compounds. And these organisms, they want to multiply, these organisms, they want to survive, so they have to use more and more inorganic compounds. They oxidize more and more these substrates, or they use more and more these substrates, and we can take the advantage of the higher activity of these organisms. They are doing for themselves, but this activity would be generated, this activity can be harnessed by ourselves, and we can get products from this, as for example by using the thiobacillus group of organism as which is requiring more amount of ferrous sulfate. So whatever amount of the biomass you will get in case of E. coli or bacillus, by using the unit of the glucose or any other carbon source which is few hundred times higher as compared to the amount of the biomass you will get in case of this thiobacillus ferrooxidans. Now it is known as acid thiobacillus ferrooxidans. So in that conditions they have to utilize more amount of the ferrous, and the more amount of the ferrous is used, ultimately the product, the more amount of the ferric is generated, and that ferric is used for the extraction of metal ions. The another example is nitrifying bacteria, where they are utilizing ammonia and oxygens, and from that they produce 132 kilocalorie energy and NO2, water and hydrogen ions. As we know that the production of hydrogen ions would be very much useful in nowadays to go for the conversion of the many compounds. Looking to the basic compounds which is required for the growth of these organisms, either it may be in the form of a organic compounds or inorganic compounds, either they are in the form of a macronutrients, micronutrients or growth factors, or in terms of various types of electron acceptors or electron donors. These types of the nutrients are incorporated in the culture media to grow these organisms. So types of the culture media which are used for the cultivation of microorganisms, they would be listed as below: the synthetic medium, complex medium, selective medium, differential medium, enriched medium or enrichment medium. Now various types of this name is given, and synthetic medium means, as I said, in case of the growth of prototrophic organisms which can grow in a minimal medium. When we talk about the minimal medium, now we are knowing that what we are adding. The minimal medium, we are adding glucose as a carbon source, we are adding either ammonium sulfate or ammonium chloride as a nitrogen source, then we may be adding magnesium or potassium or calcium as some of the nutrients. So any medium where chemical composition is completely known to the person who is preparing, then that medium is known as a synthetic medium. While the other media where you cannot know the chemical compositions, you know some of the ingredients, but you are adding some of the complex compound such as peptone, you are adding meat extracts, yeast extracts or some of the vitamins. So the chemical composition of this media is not completely known, and this type of media, they are known as a complex media. Selective medium, differential medium, enriched media and enrichment medium, we are looking in detail. Now what do you mean by the enriched medium? As I have given you the various name: enriched medium, enrichment medium, selective medium, differential medium. So what is enriched medium? The meaning of the enriched medium is a medium which contain almost all the types of nutrients, which is enriched in all the types of nutrients, full of nutrients. This type of medium is known as enriched medium, which contain complex organic substance such as blood, serum, maybe hemoglobin or maybe special growth factors or vitamins or specifically added amino acids. And in this media most of the organisms, they are growing. In case of the enriched media, most of the organisms are growing. Bacteria that require growth factors and complex nutrients are named as a fastidious organisms. So even in case of when you are preparing a medium which contain organic compound, but certain organisms which require specific addition of some nutrients, then they are known as phosphatase compound. Blood agar, which is made up by adding a sterile sheep or horse blood or rabbit blood, and which is sterilized and which is used normally for the cultivation of a pathogenic organisms. Now, how the enrichment medium differs from an enriched medium. As we have seen, in case of enriched media, most of the nutrients are present, either they are present in a complex form or they are present in a simple form, while the enrichment medium, when you want to isolate organisms from natural ecosystem and you are interested to isolate group A or group B types of organisms, you cannot separate them in a natural sample. As for example, suppose if you want to isolate Salmonella group of organism from a urine, or dysentery from a stool sample, or if I want to isolate Salmonella or dysentery or E. coli or Bacillus from a water sample or from a sewage sample or from a soil. So, these are the natural source, and where hundreds type of the different organisms are present, and it is not necessary that the type of organism what you are interested, they may be also present in a large quantity. So, specifically when you want to isolate organisms which are present in a small number as compared to other organisms which are present in their natural sources, then if you go for dilution methods, then definitely organism of your interest will get diluted and you will not get the growth of this organism.